Hello everyone, my name is Camila Arroyo-Salvo. I'm veterinarian and currently a doctoral fellow under the direction of Dr. Silvina Perez Martinez and Dr. Andres Gambini in the Biology of Reproduction in Mammals Lab in Cefibo Institute, CONICET, Argentina. Our lab has been working in understanding mammalian sperm biology in species such as human, mice, cattle, and horse. Today, I'm presenting a recent work in donkey sperm titled Phospholipase C zeta, characterization and egg activation ability of donkey sperm. Assisted reproduction in horses has rapidly increased in the last few years. This has opened the opportunity to explore its application in other equids, such as donkeys considered vulnerable or endangered. To date, no in vitro blastocyst production has been reported in donkeys. So our work aimed to describe the presence and localization of donkey phospholipase C zeta, considered to be the physiological molecule responsible for generating calcium oscillations that induce egg activation, and to compare the ability of the donkey sperm to induce the pronuclear formation and recruit maternal SMARCA4 when injected into pig eggs through ICSI. For this, frozen ejaculates from 2 yaks and 3 stallions were thought and fixed to evaluate the localization patterns of PLCζ by immunofluorescence. Additionally, stallion and donkey sperm were used to perform heterologous ICSI into pig eggs. 18 hours after ICSI, the presumed zygotes were fixed and stained with TAPI and SMARCA4 antibody for pronuclei visualization. 4 different main patterns of PLCζ localization were observed by immunofluorescence after analyzing a total of 590 sperm cells. F, flagellum only, A, acrosomal, PA, post-acrosomal, and A+PA, acrosomal and post-acrosomal. Surprisingly, around 60% of donkey sperm showed flagellum and acrosomal PLCζ localization, whereas in horses, post-acrosomal and acrosomal plus post-acrosomal were the most common patterns, around 80%. All sperm cells showed flagellum localization of PLCζ. Interestingly, pig oocytes were significantly less activated after injection with donkey sperm than horse sperm. However, SMARCA4 pronuclear localization levels in the zygotes were similar among groups. So, our results describe for the first time the localization patterns of PLCζ in donkey sperm. We observed that horse sperm has a significantly increased ability to induce pig oocyte activation compared to the donkey. Since PLCζ is a crucial molecule for oocyte activation, we hypothesized that the different patterns of localization described for donkey sperm could be associated with low activation rates observed when injected into the egg. Therefore, strategies to improve the in vitro development of donkey ICSI embryos, such as assisted activation, might be required. Of course, more research is needed to confirm these preliminary results. Gracias.